Question number 1. The insertion of a gene of interest through a vector and which is replicated independently in Escherichia coli is termed as, choice A is rDNA technology or gene cloning, B transduction, C replication, D plant tissue culture. Whenever we take a gene of interest, any gene of interest taken and loaded into a vector. A vector primarily could be a sort of a plasmid. A plasmid is a circular molecule and you are taking your gene of interest and placing it into the circular molecule. When you are loading it into a vector and this gene and the plasmid is capable of independent replication, it is capable of making multiple copies. Within a bacteria, that is within Escherichia coli, if the gene of interest is loaded into a plasmid and the plasmid is able to make n number of copies, then the entire process is primarily termed as gene cloning, or it is also termed as recombinant DNA technology or rDNA technology. Now, what exactly is the meaning of recombinant DNA technology? We are making a recombinant DNA molecule. That is, you have a gene of interest which is loaded into a plasmid. A plasmid is again made up of a DNA and it is integrated within the DNA. So, you are making a chimeric gene. A chimeric gene is made up of several components of genes. So, we have a recombinant DNA, and because we are making multiple copies, a clone refers to any multiple copy of a small fragment of the DNA in which there is a gene of insert. If you are making multiple copies, say 1000 copies of a particular sort of a gene within a bacteria, then the term which is used for that is termed as gene cloning. So, in recombinant DNA technology or gene cloning or genetic engineering, the basic facts are: first, we need to isolate the gene of interest, B, we need to load it into a plasmid, C, the plasmid should be able to make multiple copies of itself, that is circular molecules. The plasmid acts like a vector and the vector is situated within the host which is called the Escherichia coli, and from there the gene of interest is transferred from one plant to another plant. While if you take transduction, this choice number 2 is termed as transduction. Transduction primarily relates to naked DNA transfer. Naked DNA transfer between two bacteria are termed as transduction. We have got primarily conjugation, transduction and transformation. And C, the term replication refers to DNA replication, that is how DNA makes double copies of itself. And the choice D refers to plant tissue culture. Plant tissue culture is aseptic growing of any particular part of either a cell, a tissue or an organ in a defined medium under controlled conditions. So, given the choices, whenever we have a gene of interest via vector, replication independent into Escherichia coli, the popular term which is used for this is termed as rDNA technology or gene cloning. Otherwise, it could also be referred to as genetic engineering. The answer is A. Question number 2. The first steps towards the generation of DNA fragments from genomic DNA is by A, vectors, B, restriction enzymes, C, cloning, D, gene expression. If we ever have a genomic DNA, the genomic DNA refers to the total DNA content of the organism is termed as a genomic DNA. It is something like a sort of a noodle pack which is present within a sort of a plant cell. So, when you take the genomic DNA, from the genomic DNA, if we want to isolate our gene of interest, it is like isolating a needle in a haystack. So, after we isolate the gene of interest, the gene of interest is isolated via a use of restriction enzymes. Restriction enzymes can cut the sort of DNA at specific sites. Then the gene of interest is loaded into a sort of a vector like a plasmid vector or a bacteriophage vector. Then later on there are multiple copies of the genome or the particular gene of interest is made, and then there is a sort of a transfer or the genetic transformation of the gene occurs into the host cell, and thereby the entire process of gene cloning or making multiple copies is made. So, the first steps primarily involves, that is, it involves isolating the DNA, cutting the DNA, loading it into a plasmid, and again making multiple copies of it, and the expression and the transformation of it. So, all these 6 steps primarily with relation to isolation of the DNA, then again cleaving the sort of a DNA by restriction enzymes, C, placing the gene of interest into the sort of a plasmid or the vector, D, expression of the sort of a vector in the E. coli, and E is taking the sort of a gene of interest and again loading it into the host cell and enabling genetic transformation. All these are continuous steps in the broad technique of gene cloning. So, when you take choice number 2 is by A is vectors. Vector primarily refers to any particular sort of an organism or a sort of a molecule of interest which can take in your foreign DNA is termed as a vector. A vector will shuttle only from one host to another host. Like a common vector is, if you take PBR322 is a vector, or else you take cauliflower mosaic virus as a vector. Otherwise, BUC is a back vector or bacteriophage is a vector. And when you take B, restriction enzymes are enzymes which are termed as molecular scissors which can cleave the sort of a DNA at specific sites. The term cloning here refers to making all the sort of sequences put together and making multiple copies of the gene of interest. So, it is just like you are xeroxing a sort of a gene of interest and making 1000 copies of it. So, that process is termed as cloning, and cloning involves all the steps together. The term D, gene expression, relates to how does DNA make RNA and how is RNA seen in the form of a protein. So, the expression, that is how DNA to RNA to proteins is termed as gene expression. So, when you want to generate DNA fragments from a genomic library, the right answer refers to as cloning. Cloning refers to it's a huge process or it's a big process which involves several steps, and ultimately you are getting multiple copies of your gene of interest. Hence, for answer 2, the answer is C. So, when you take gene cloning, in gene cloning, primarily we have to isolate the DNA. Then second, we need to cut the DNA by restriction enzymes. Third, we need to transfer the sort of a DNA, transfer the gene of interest into a sort of a vector, that is a sort of a plasmid. Fourth, the plasmid will be asked to make multiple copies. Then the integrated gene that is your foreign gene will combine with the sort of a host DNA, integrated gene will be able to express. And lastly, you will be making the process of cloning, that is you will be making multiple copies of the gene of interest. So, all these are the basic steps related to steps in gene cloning. Hence, when you take question number 2, the right answer is C, which relates to directly with relation to the with the term cloning. Question number 3, Smith, Arbour, Nathans, 1978 were awarded the Nobel Prize for discoveries on A, HindIII enzyme, B, restriction digestion, C, endonucleases, D, all the above. When you take 1978, primarily there were 3 important scientists whose names were Smith, Arbour and Nathans. They had worked with relation to one particular restriction enzyme called HindIII. HindIII was the first restriction enzyme which was discovered in Escherichia coli, and primarily this particular restriction enzyme could locate a specific sequence in the entire sort of a DNA. It could locate a specific sequence in the entire sort of sequence in the entire DNA, and it could remove the sequence. So, when the restriction enzyme cuts the sequence specifically, the term which is used for this is termed as restriction digestion. So, restriction digestion primarily means cleaving. So, it's a cutting of the sort of a sequence of interest, and this cutting is attributed to a set of enzymes which are termed as endonucleases. So, under the broad category of endonucleases are enzymes which are within the nuclease. They can cut the sort of DNA segments. So, under the broad category of endonucleases, we have HindIII. HindIII is a restriction enzyme. Under endonucleases category is restriction enzymes, and these restriction enzymes can perform restriction digestion. So, the Nobel Prize for Smith, Arber and Nathans had been given for the discovery of endonucleases, specifically HindIII, by identifying the restriction enzyme which causes restriction digestion with relation to specific sequences. Hence, for question number 3, the answer is D. The right answer is D. Question number 4, match the following. We have got two columns, column 1 and column 2. Under column 1, we have got A is ECOR1, B is BAM-H1, C is TAG1, D is HindIII. Under column 2, first one is Haemophilus influenza, 2 is Thermus aquaticus, 3 is E. coli, 4th is Bacillus amyloliquefaciens. The choices are A is A3, B1, C2, D1, B is A3, B2, C1, D3, C is A4, B3, C4 and D2, and D is A1, B1, C3 and D4. Now, when you look at the sort of choices, when we take Escherichia coli, ECOR1 is one of the things. Next, we have got BAM-H1. Third one we have is TAQ1, and next we have got is HindIII. ECOR1 is always primarily associated with Escherichia coli. If you recollect, when you take the nomenclature of restriction enzymes, the first one relates to the genus and the next two letters relates to the species. So, Escherichia coli, ECOR1 relates to Escherichia coli. Similarly, when you take BAM-H1, BAM-H1, it relates to, it has been isolated from Haemophilus influenzae. And when you take TAG1 relates to primarily from, this has been discovered from a microorganism which grows at very high temperatures, that is Thermus aquaticus. Now, for certain other things like TAQ, it relates to TAG, whereas for this particular thing BAM, this is an older name, so the new name is Haemophilus. And similarly, when you take HindIII-D relates to primarily, it is also a sort of organism which has been discovered primarily from Haemophilus. So, when you take the sort of choices, the right choice is A3, ECOR1 with relation to Escherichia coli, B1, that is you got BAM-H1 with Haemophilus influenza, then C is 2, that is TAG1 is isolated from Thermus aquaticus, and D is primarily HindIII is also from Haemophilus influenza. The most important enzyme here is, the most popular sort of a restriction enzyme is ECOR1. The first or the foremost restriction enzyme in the entire world of molecular biology is Escherichia coli. And a very important enzyme which is also a basis for polymerase chain reaction or for amplification of the DNA is derived from TAG1. TAG1 is a sort of a restriction enzyme which is extracted from Thermus aquaticus, which is a very important bacteria which grows in hot springs. So, at the highest temperature it can denature and also renature bacteria, that is the DNA can both be cleaved as well as it can also be sealed. Hence, among the sort of choices, the right choice is answer is A. Question number 5. A suitable gene cloning vector is one which has A, small and it has got high copy number, B, has been naturally present in E. coli, C, with antibiotic resistant genes and an ORI, and D choices only A and C. Now, when you take any gene cloning vector, or when you are attempting gene cloning, that is making multiple copies of a particular gene of interest, and you require a vector which can transfer the sort of foreign gene into the bacterial cell and again back into the sort of plant cell, what are the characters of the gene cloning vector? Or what are the ideal properties of a gene cloning vector? Whenever we take any gene cloning vector, first thing is it should be small in size in terms of kilobase space. The reason it should be small in size is because it should be able to take the load of the DNA and move within the bacterial cells and between the host and the recipient cells. So, the smaller a vector, the more easier it is to manipulate, whereas the bigger vectors or very large vectors are difficult to manipulate. So, first important attribute, it should be small in size. And next one is it should have high copy number. That is, it should be able to make multiple copies of itself. Making multiple copies of itself is termed as relaxed state. When you take stringent plasmids, or stringent means they cannot make multiple copies of themselves. So, if you got a bacteria and within a sort of a bacteria you have a plasmid is an extra chromosomal DNA. The extra chromosomal DNA is circular. And if you want to make multiple copies, supposing it is able to make 20 copies of itself, that means it has got a high copy number. Whereas a plasmid has got makes only 2 copies of itself, it has got low copy number. So, a small size and a high copy number are very important attributes of any particular gene cloning vector. Now, along with this, any plasmid should have an ORI, that is, it should have a site of origin. So, a typical plasmid will have an ORI, it should be small in size and it will have high copy number. Then in addition to this, it would always be having antibiotic genes. These are termed as antibiotic genes. That is, it will be resistant to antibiotic genes. So, supposing you are taking a culture and treating them with relation to an antibiotic, it will be antibiotic resistant, that is, it will be protected. And very important thing is it should have an ORI. Only when it has an ORI, it is capable of replication. So, presence of antibiotic resistant genes. So, what are the common antibiotic resistant genes? If you take AMPR, ampicillin resistance, as well as tetracycline resistant genes. So, presence of antibiotic resistant genes, presence of an ORI, being in a high copy number or a relaxed state, and being small in size. These are the basic attributes of the most suitable vector which is used in genetic engineering or in the case of biotechnology. So, among the choices we have the choice D says only A and C, and the answer is D. Question number 6. The specific nucleotide sequences on the DNA which are cleaved by restriction enzymes are termed as A. cohesive ends, B. palindromic DNA, C. sticky ends, D. restriction sites. Supposing we have a DNA and the DNA is made up of nucleotide sequences. Among the nucleotide sequences, if there are specific nucleotide sequences, these specific nucleotide sequences are recognized by restriction enzymes. So, this specific recognition is termed as recognition specificity. So, recognition specificity is a characteristic feature of restriction enzymes. So, because of the property of recognition specificity, the restriction enzymes will identify specific nucleotide sequences. Like if you take restriction enzyme 1, it will identify A, C, D, E and G. If you take restriction enzyme 2, it will identify B, then it may identify H, then it will identify Z. So, they recognize specific sequences, and these specific sequences which are identified, these are the places where the enzyme is going to cut the sort of a DNA sequence. So, because it can cut the DNA sequence, these regions are primarily termed as restriction sites. So, restriction sites are the cutting planes on the DNA which is recognized by restriction enzymes. So, among the choices, the right answer is D. However, when you take the recognition sites, in the recognition site, what is the most important characteristic feature? The most important characteristic feature is when you take the nucleotide sequences are mirror images of each other. That is, if you take a DNA and if you take another DNA, both of them will show palindromic sequences. When you say palindromic sequences, it means if you read the word Madam, I am Adam. So, here when you read it from here, it is MADAM, I am ADAM, whereas when you read it from here again, it is Madam, I am Adam. So, these are termed as palindromic sequences. So, similar palindromic sequences are present on the DNA. So, these are the sequences where the cutting occurs, but the question asks which is the place? The place is a restriction site, hence the answer is D. Question number 7. In transgenic rice, the gene therapy includes A. increased vitamin A content, B. more of carbohydrates, C. early maturity and tastier rice, D. increased shelf value. When you take transgenic rice, it is the rice which has been developed by transgenic technology or genetic engineering. It has been primarily studied by Porteous. Now, when you take Porteous primarily was a scientist from Switzerland, from Zurich, who was interested in fortifying rice. That is, he was the first person to develop fortified vitamin A rice. That is, normally in order to remove blindness or to remove deficiency due to lack of vitamin A in developing countries as well as in less developing countries where the staple food has been rice, it is the most important objective that rice should be fortified or it should be enriched with vitamin A. So, primarily in Zurich, Potters primarily developed transgenic rice. So, what happens in the case of the transgenic rice? In transgenic rice, 3 genes, 2 from a bacteria and another 1 gene from daffodil plant has been synthesized together. 3 genes have been synthesized together and all the 3 genes are led to altering a sort of a pathway. They could control a pathway of beta carotene. So, genetically the 3 genes were engineered to secrete beta carotene, and beta carotene is primarily secreted in the sort of an aleurone layer. So, beta carotene is a sort of a precursor for vitamin A. So, synthesis of beta carotene involving 3 genes of plants primarily led to the development of fortified vitamin A, that is vitamin A was fortified or enriched. So, there was enriching of the rice plant so that when people consume rice they could get vitamin A after digestion. So, this is primarily termed as transgenic rice. So, transgenic rice is always related to vitamin A in the case of rice content. Hence, the answer is A. Question number 8. The molecule which has been experimentally manipulated in the lab and contains novel or modified genes is called A. cloning vector, B. recombinant DNA, C. molecular cloning, D. cloning host. When we study genetic engineering primarily relates to manipulation or modification of the gene of interest. So, modification of a gene of interest which occurs in the lab is primarily termed as in vitro. So, when we perform any experiments in the lab, it is termed as in vitro and it is experimentally manipulated. So, when you experimentally manipulate a particular sort of a molecule, so in this case it is a sort of a DNA molecule. When you manipulate a DNA molecule and thereby you either modify it in such a way that it has got novel genes, novel means new, it has got novel genes or it has modified genes. Like common examples could relate to A. early flowering, high nitrogen fixing ability, high photosynthetic efficiency, then again maybe high amylose content or increase like softening of tomatoes or enriching rice with vitamin A, otherwise maybe high gluten content in the case of maize or more tastier apples and fruits. So, all of these are primarily because of the presence of new genes or new proteins. So, we have normal modified genes and proteins especially with relation to manipulation of the molecule. So, this technique is predominantly termed as recombinant DNA technology. Now, when we take the choices A, cloning vector, a vector is a molecule or a sort of a piece of the DNA which can transfer the sort of a DNA from one organism to another organism. It is nothing but a sort of a transport vehicle. When you use the term recombinant DNA means we are trying to study under in vitro conditions, experimentally manipulate the molecule for getting novel modified genes. Whereas, when you take molecular cloning is the tool and the technique. Molecular cloning refers to the entire tools and techniques in gene cloning. that is the methods and the processes which occur in the lab, is termed as molecular cloning. Cloning host refers to what is the name of the plant. So, when we are dealing with relation, the most important word here is how to produce novel modified genes. Like if you want to improve a particular crop for climatic change, or for high fertilizer response, or for stress tolerance, or supposing the soil is not enriched by means of nutrients and you want a plant to grow, you want a plant to be adapted, or you want higher vitamin A content in a particular sort of plant, then biotechnology seems to be the sort of an answer. So, the answer here is restriction recombinant DNA, and the choice among these is answer is B, that is recombinant DNA technology. Question number 9. A collection of cloned fragments together representing the complete genome of an organism is called A. recombinant DNA, B. cistron, C. gene, D. genomic library. As you understand, when you say clone, a clone refers to a sequence of DNA which has the gene of interest. Which has been manipulated. And which are identical. Again, which can be multiplied. So, when we are doing gene cloning, our objective is to isolate the gene of interest and make multiple copies of it, multiple identical copies of it. So, if we take a sort of a plant DNA, or you take a bacterial DNA of the entire sort of a genome, you take the complete genome of an organism, maybe you take Escherichia coli, or you take a human being, otherwise you take even rice. So, from rice, if you take the entire DNA sequence, from the entire DNA sequence, you isolate the sort of a pieces. You take the genes of interest, maybe like if you take the alphabets from A to Z, if you think if a gene of interest is say D, F, Z and say Q, we isolate those particular sequences, and after you isolate those particular sequences of gene of interest, you place them in Petri plates. These are your genes of interest. So, after you isolate the gene of interest, you could load all the sort of a Petri plates, maybe on a sort of a shelf, which may amount to about some 10,000 to 20,000 of these. So, 10 to 20,000 of these could primarily represent the entire sort of a genome of the organism, and this collection of cloned fragments, the collection of cloned fragments of the entire genome. So, you are representing the entire genome, you are taking clone fragments. When you say clone fragments, it means identical fragments. You are aiming to multiply them and also manipulate them. So, this primarily is termed as the genomic library. So, a genomic library represents a collection of all the sort of a clones. A collection of all the clones or the identical DNA of organisms, identical DNA or the gene of interest of organisms, which truly represents the genome. Hence, the answer here is D. Question number 10. In vitro regeneration of plants from organs, tissues, cells or protoplasts, and true to type selection is otherwise termed as A. Anthoculture, B. Micropropagation, C. Organogenesis, D. Callus. When we are aiming at regeneration of plants, when you say regeneration, it means making multiple copies of the plants, multiple copies of the plants which are identical. When we want to regenerate, making multiple copies in large numbers very quickly for commercial purposes and we are practicing in vitro techniques, that is plant tissue culture techniques, and our tool is either the protoplast or the cell or the organ or the tissue. And here whenever we are regenerating, all the parts, the selection is true to type. When you say true to type, they morphologically they are identical to the parents. So, this method of propagation is primarily termed as micropropagation. Now, why should you have micropropagation? Micropropagation is a method wherein you can grow orchids, bamboo, mango, sugarcane and any medicinal plant in large numbers. You can grow them in very large numbers, make multiple copies of it, and because they can be grown together to make multiple copies, you can make, say, from one single cell by micropropagation, you can easily establish about some 100 plants which relate to teak. The most important thing is they are all pathogen free. That is, they do not have viruses. So, micropropagation is one method wherein you can get pathogen free material and they are true to type. Because they are true to type, it is very useful for reclamating large lands. When if you want to reclamate the deserts or you want to reclamate wastelands, otherwise you want to build a sort of a barrier primarily with relation to winds and also with relation to any sort of a natural calamities, and you want all the plants to be of the same type, then you primarily proceed by the technique of micropropagation. So, micropropagation is a very important technique, especially with relation to multiplication of spices, condiments as well as teak and orchids. Now, here the right answer is B. Question number 11. The culture of carrot tissue and callus formation indicates A. Totipotency of plant cells, B. Genetic engineering, C. Fermentation, D. Gene manipulation. Whenever we have a single cell, from a single cell, if we have to regenerate and it forms the whole plant, this inherent property of plant cells is termed as totipotency. So, according to the concept of totipotency which has been proposed by Haberland. Haberland had been the father of plant tissue culture, as well as it has also been worked out by F.C. Stewart. F.C. Stewart is a world famous plant physiologist who has worked on carrot, and he could generate the entire carrot tissue from a single cell, and in the process it could go to form the sort of a callus. A single cell could give rise to the callus. The callus on differentiation can give rise to the entire plant. So, this property is primarily termed as totipotency. Now, totipotency is a unique characteristic feature of all plant cells. It is analogous to Ravana's head. When you cut one, you again regenerate the other one. So, totipotency is a special characteristic feature of plant tissue culture. So, given a medium, you can isolate a single cell. From a single cell, you can grow the entire plant. Whereas, genetic engineering primarily relates to manipulation of a single gene of interest. Fermentation is a method of anaerobic oxidation, and gene manipulation primarily relates to how do you take a gene of interest and you add either a sort of a promoter sequence, or you have a repressor sequence, or you have an expression sequence. So, the most important fundamental basis of all plants is the property of totipotency, and the answer is A. Question number 12. The method of growing isolated cells aseptically and culturing on a nutrient medium is called A. Tissue culture, B. Organ culture, C. Cell culture, D. Callus culture. When we take cells, and especially the cells are isolated cells, single cells, we take single cells and grow them aseptically, that is, aseptically means primarily without any sort of a contamination, and you are culturing on a nutrient medium. So, a single cell culture is normally called as cell culture. Among the choices, we have A is tissue culture. Tissue culture relates to the culturing of either a single cell or a group of cells or a tissue aseptically, whereas when you take an organ relates to an anther, whereas a cell relates to an individual cell, and a callus is an unorganized mass of tissue. So, when we are taking single cells, a single cell culture primarily is an example of an isolated cell culture. So, an isolated cell culture is the basis for primarily for batch culture, which is useful in the production of secondary metabolites. Like if you want to get a cancer curing drug, for getting a cancer curing drug, we have to take a particular cell and you will have to grow it in either a batch culture or in a suspension culture. So, always remember, single cells means isolated cell culture. So, a cell culture is a very important term, and the right answer here is for 12 is answer C, which primarily relates to a single cell culture or a cell culture. Question number 13. A gene bank is an example of A. Ex situ conservation, B. In situ conservation, C. Old adaptive varieties, D. Wild relatives and ancestors. Now, when you take a gene bank, the meaning of the term gene relates to DNA, and the meaning of the term bank is it is helpful in conservation. We are conserving a sequence of the DNA. So, whenever we conserve anything in ex situ means outside their natural habitat. Like in case, instead of in the tropical rainforests, we take and we only culture and maintain seeds. It is an example of ex situ conservation, only seeds. Or we take only the sort of a plant and we maintain them in the field. It is termed as a field gene bank. So, we have a seed bank, then we have a field seed bank. Otherwise, we primarily maintain them in botanical gardens. A plant maintained in botanical gardens is also an example of ex situ conservation. Whereas, when you take in situ conservation, in situ relates to in the habitat. Like if you take a sanctuary or if you take a protected area, if a plant or an animal is present there in its own habitat, it is termed as in situ conservation. old adaptive varieties cannot constitute a gene bank, because a gene bank, the objective of gene bank is you need to conserve the DNA and utilize it for any other useful purpose at a later period of time. It will not just be a sort of a collection of wild varieties and ancestors. Hence, when you say gene bank, primarily it relates to ex situ conservation or outside the natural habitat. Hence, the answer is A. And it includes A, seed bank, B, field seed bank, and C, it includes botanical gardens. Question number 14. Guha and Mukherjee are Indian scientists famous for A, green revolution, B, anther-culture and haploids, C, genetic engineering of rice, D, embryo culture. If you take Sipra Guha and Mukherjee from the University of Delhi have been pioneers with relation to the technique of anther-culture. It is the experimental manipulation of unopened anthers which can be cultivated in vitro on a nutrient medium aseptically under controlled conditions wherein if you take the anthers and grow them in plant tissue culture, from the anthers we can get, it can lead to the production of haploid plantlets. Now, haploid plantlets are very important because we will get primarily anthers which are haploid. So, they will have n number of chromosomes and you can easily double them, so that you can get homozygous diploids. So, the very important application of this is production of haploid rice. So, Sipra Guha and Mukherjee are Indian scientists famous for anther-culture and haploids. The answer is B. Question number 15. Skoog and Miller are famous for A, the concept of hormonal control in organ formation, B, one gene, one enzyme hypothesis, C, operon concept, D, human genome project. If we look at Skoog and Miller, they are primarily people who have worked relation to plant tissue culture. And they primarily established that the relationship between auxins, gibberellins and organ formation. Whenever there is a balance between auxins and gibberellins, if it is equal, then it always relates to the formation of callus in a medium. So, they were the scientists who primarily suggested that organ formation relating to either high auxin content or high maybe low gibberellin content or even cytokinin content, it relates to the relationship between hormones and organ formation. So, Skoog and Miller were primarily identified that the formation of the shoot or the formation of the root was specially controlled by the levels of auxins and cytokinins. The auxin cytokinin ratio determines the formation of shoot and root in plant tissue culture? The right answer is A. Question number 16. In gene cloning, for transfer of DNA fragments from a gel to a membrane which is called nitrocellulose, the technique is A. Northern blotting, B. Southern blotting, C. Restriction digestion, D. DNA replication. Whenever we want to practice gene cloning, initially we have the DNA. The DNA has to be isolated. After we isolate the DNA, from the DNA we take our gene of interest, which is termed as a clone. Now, to identify this or to transfer this sort of a DNA, we need to take this sort of a DNA and load this particular DNA fragment into an electrophoretic system which is termed as polyacrylamide gel electrophoresis. So, this is a method of electrophoresis. So, the DNA molecule, because it is amphoteric, because it has got plus and minus charges, when it is subjected to electrophoresis, it will migrate in the form of bands. The separation of the DNA bands occurs on the basis of its molecular weight. So, after we separate this sort of a DNA, this particular DNA band is loaded onto a membrane. This is a nitrocellulose membrane, just like a nylon mesh. So, you take the nylon mesh and you place it on the DNA. Then it can take images of the DNA. So, it is a nitrocellulose mesh. Now, after you take this particular nitrocellulose mesh, the same sort of a DNA profile can be edited and you could locate your gene of interest. Now, this technique of transfer of the DNA, how do you transfer the DNA? It's a DNA transfer technique onto nitrocellulose, has been studied by a scientist whose name is Southern, and because it involves blotting, it is termed as Southern blotting technique. So, the Southern blotting technique is a method of transfer of the DNA. Hence, among the choices of northern blotting, southern blotting, restriction digestion and DNA replication, the right answer is southern blotting, that is, the answer is B. Question number 17. The source of carbon in plant tissue culture is A. Auxin, B. Sucrose, C. Pectin, D. Amylose. Whenever we take plant tissue culture, all plant tissues primarily require a medium, and the medium is termed as a nutrient medium. And the nutrient medium need to be aseptic and it should be autoclaved. That is, it should be subjected to very high temperatures so that it becomes very sterile. Again, a typical medium should have primarily agar so that it does not melt and it solidifies. In addition to this, the nutrient medium will be neutral and it will have a constant sort of a pH. Now, if a plant has got to grow, if a small sort of a an eggplant has got to grow, or maybe a part of a root, shoot or a leaf should be grown under artificial conditions, the first very important component for it is carbon. Along with carbon, we also have hydrogen, oxygen, nitrogen, sulfur and phosphorus. So, the source of carbon is predominantly in plant tissue culture is sucrose. because sucrose is a very important carbohydrate which again can get broken down to form fructose and other compounds, and normally it is a stable compound. Whereas when you take auxin, auxin is a growth hormone. Pectin primarily exists as a sort of molecule which is present as calcium pectate in the sort of middle lamella, and amylose is a storage product. So, if we want a metabolic product and which again which is complex, at the same time which is stable and which is capable of breaking down to form simpler products, the choice is sucrose. So, sucrose is the major source of carbon, especially in the case of plant tissue culture, and the right answer is B. Question number 18. For in vitro rooting of shoots in plant tissue culture, the growth hormone used is A. auxins, B. cytokinins, C. gibberellins, D. ethylene. Now, if we take shoots, we want to produce in vitro, we take the plants and we are interested in rooting. So, the object here is, if we want to develop rooting of shoots, which is the growth hormone which is required here. Now, always whenever you want rooting, we need to primarily give more of auxins, high auxin content and low cytokinin content results in rooting. Whereas, if we want shoots, high cytokinin content and low auxin content gives rise to shoots. So, it should always be remembered that cytokinins promote cell division. So, because they promote cell division, it results in the formation of shoots. Whereas, when you take high auxin, auxins causes cell expansion. So, roots are very important, especially for establishment of either the explant or the first sort of a cell or the plantlet. So, in order that the plantlet or the explant form roots, always high auxin and low cytokinin. So, among the choices, the right answer is A. Now, it is very important that the question which has been framed refers to in vitro rooting of shoots, in vitro rooting of the shoots. It is not shoots, it is in vitro rooting of the shoots, is primarily promoted by the primarily due to high auxin content here. Question number 19. Agrobacterium tumefaciens is called A. Human Genome Project, B. Phage DNA, C. Plasmid, D. Nature's Genetic Engineer. Now, when you take Agrobacterium tumefaciens, it is a soil negative gram negative bacteria. It is normally associated with both dicots and monocots, and Agrobacterium tumefaciens has a very important plasmid called a tumor inducing plasmid or a TI plasmid. The Agrobacterium tumefaciens causes a disease which is termed as crown gall disease. Now, when you take Agrobacterium tumefaciens, in its cells which are present in the roots of plants, it has inherently a TI plasmid. The bacteria Agrobacterium tumefaciens has a resident plasmid, that is, the plasmid is by nature associated with the Agrobacterium tumefaciens. So, this particular plasmid has got the capability of taking its own DNA if you take this is the T, if you think this is the Ti plasmid and if you think this is the DNA, it is capable of taking its own DNA and integrating within the plasmid. So, in nature normally it cuts the DNA and takes its own DNA and loads into the plasmid, and this particular plasmid later on it can easily be transferred to another sort of a cell. Now, when you take this Ti plasmid, the Ti plasmid has got its own ORI, it has got its own origin of replication, and primarily it can take in a foreign DNA or a foreign gene of interest all by itself without the artificial genetic engineering. It can take a foreign gene from the host plant. It can isolate a foreign gene from the host plant and it can load it into the sort of a plasmid. Hence, Agrobacterium tumefaciens is termed as nature's genetic engineer. So, somewhere in the 1980s, when you take the technique or the start of recombinant DNA technology or plant biotechnology or primarily gene cloning has started with Agrobacterium tumefaciens. This is the most suitable sort of a vector for plant genetic engineering. Due to the presence of Ti plasmid, due to its association with both monocots and dicots, and because it can easily, the Ti plasmid can easily result in integration of the foreign DNA, and again it can easily be expressed and it can also be cultured. So, these are the 5 reasons due to which you call it as a sort of a nature's genetic engineer, and the answer is D. Question number 20. Amplifying DNA fragmentation involves A. Restriction enzymes, B. Gene conservation, C. Embryo culture, D. Polymerase chain reaction. Now, when you take a gene of interest, we have the technique of A. Gene cloning, and in gene cloning from a huge directory of the sort of a DNA, we isolate our gene of interest. And once we have an isolate the gene of interest by restriction digestion, by placing it in a plasmid, by performing genetic engineering, we get our sort of a clone gene wherein we have multiple copies of the particular sort of gene. Now, with the same piece of the gene, we want to amplify it, make multiple copies of it without using a probe or without using any other sort of restriction enzymes. There is one particular technique which is termed as polymerase chain reaction, PCR. Now, when you take polymerase chain reaction, it is based on the principle of DNA replication. And it has been studied by a scientist whose name is Mullis. So, when we have a DNA, a small bit of the DNA, this DNA bit, to the DNA bit you add a probe. So, when you add a probe, the DNA can both anneal, that is join, as well as it can also denature, that is separate. So, when you take the sort of DNA and you subject it to a spin, you subject it to several rounds of DNA replication by causing alternate heat and cold. when you subject it to very high temperatures and very low temperatures, the DNA can both separate as well as join. By subjecting it to several cycles of thermocyclic reactions, the DNA can get both separated as well as joined. Thereby, you make multiple copies of your gene of interest. So, you will be getting about some at least 10,000 copies of the gene of interest within half an hour. So, this method, this technique of amplifying the DNA of gene of interest is termed as polymerase chain reaction. And because this has been primarily discovered by Mullis, who got a Nobel Prize for this, and this is an alternative route instead of using biochemistry. If you quickly got your sort of gene of interest, say you got a gene of interest for curing cancer, or you got a gene of interest for which controls diabetes or hypertension, or you got a gene of interest primarily with relation to curing brain tumor, then that particular gene can easily be amplified. So, it is a technique which is the basis for a gene sequencing. And it is a technique for gene mapping, for determining the map, or it is the basis for the Human Genome Project. The right answer among the choices, answer is D, that is PCR.